Now that we've learned how DNA is structured and how it copies itself, let's talk about how DNA is used to make proteins. So this is called the flow of genetic information. All right. So the DNA basically specifies traits by dictating a process we call protein synthesis. The chain of command in this whole process is DNA in the nucleus is copied into RNA, and the RNA in the cytoplasm directs the synthesis of the protein. These two processes, one in the nucleus, one in the cytoplasm, are called transcription and translation. Transcription is the synthesis of RNA under the direction of DNA. It is basically making a copy of a DNA, a complementary copy of a DNA sequence that codes for a protein. Translation occurs in the cytoplasm, and it is using that copy, that template, to translate or change from the language of bases, or DNA, into the language of amino acids, which is the language of proteins. Here we have DNA in the nucleus. The DNA is transcribed, the sequence of DNA is transcribed into RNA, which then leaves the nucleus and travels to the cytoplasm to be made into the protein. So the real secret, the code here, the sequence of nucleotides, is providing the code for making the protein. We have to convert that nucleotide sequence into an amino acid sequence, and so basically transcription is going to rewrite the DNA. The script part of the word means writing, and so we're rewriting DNA code into the RNA. Using the same language, is still the nucleotide language, but we're just rewriting it in a different way, kind of making a copy of it. It's sort of like a molecular Xerox machine. All right. The information from gene to protein is based on what's called a triplet code. The instructions are in sequences of 3 bases. These are 3 bases that don't overlap each other. They kind of make a word, and the word is called a codon. Each codon represents an amino acid. When we move from the nucleus to the cytoplasm for translation to occur, that's going to involve changing from language of nucleotides to the language of amino acids. And each amino acid is going to be specified by a codon. So here we have transcription of the DNA. Here's our DNA original sequence, and here's our complementary copy and messenger RNA, and then each group of 3 nucleotides in the RNA codes for a particular amino acid, and that goes together to make a protein. So the way this works, okay, there are 3 nucleotides that code for one amino acid. This makes, since there are 4 letters and they're grouped into groups of 3, that means there are a total of 64 possible codon combinations. 61 of those correspond to amino acids, and 3 of them are what we call stop codons. The codon AUG in the messenger RNA codes for the amino acid methionine, and it also signals the start of transcription. And then there are 3 different stop codons that act like periods at the end of sentences. Here we see the DNA code, the genetic code. This is actually messenger RNA code, and the way you read this is you find for the first base in the codon, you find it which row it's in, U, C, A, or G, as you see here. The second base tells us what column it's in, and then the third base tells us which row within that box. So if we're trying to decode UCA, our first base is U, so it's going to be in this row. Our second base is C, which means it's in this column, and so it's going to be within this box here. And then A is my third base, and that tells me that that's UCA, which codes for the amino acid serine. You do not have to worry about learning the names of all of these. If you can recognize the abbreviations, that's fine, but all you need to do is find the code in the chart, and that tells you what amino acid goes in that position in the polypeptide that's going to be built. Here's methionine, which is our start codon, and here are our 3 stop codons. Most amino acids have from 2 to 4 or even 6 codons that code for them. There are a few that have just one codon. Methionine is one, tryptophan is another, and the others all have at least two. So here's our strand of DNA to be transcribed. We're going to unzip, and we're going to use the darker blue here as our original. Then our complementary copy in messenger RNA in pink here. Notice it's the same as the opposite side of the DNA molecule, but it substitutes U for T because uracil is in RNA and thymine is in DNA. And then finally, each one of those three groups of three is going to code for a protein, a particular amino acid, and here's my stop codon at the end to stop the transcription or translation. So now that we've learned how to read the code, let's look at the process that makes this occur. In order to transcribe the RNA molecule from the DNA template, we have a process that is similar to what we saw in replication. There is a process that's going to unzip the DNA molecule and then use RNA nucleotides to match up to the DNA sequence rather than DNA nucleotides. This involves an enzyme called RNA polymerase. RNA polymerase basically unzips, copies in messenger RNA, and then re-zips the DNA back up. There are specific places of sequences of nucleotides on the DNA that mark where transcription starts and finishes, and there are also factors that signal when something needs to be made. Here we have the RNA polymerase molecule basically unzipping the DNA strand right here, plugging in the appropriate matching nucleotides, and then re-zipping the DNA back together. We call the DNA copy that is made of, RNA copy that is made of DNA, messenger RNA or mRNA. It's the original copy, basically, that signifies the sequence of amino acids that need to be put together to make the protein. It occurs in prokaryotes, it occurs just right there in the cytoplasm because remember prokaryotes do not have a nucleus. But in eukaryotes, the messenger RNA has to leave the nucleus. And the RNA is also put together a little bit differently in eukaryotes than prokaryotes. In eukaryotes, there are two parts of the DNA, or the RNA. Those are introns and exons. The exons are the coding region of the gene, and the introns are pieces in between the exons that separate them from each other. The introns need to be removed. So it's going to the messenger RNA is going to undergo a processing called RNA splicing. It's going to remove, there are enzymes that will remove the introns and then join the exons back together to produce a continuous sequence for the coding of the protein. And then there are also a cap and a tail of extranucleotides that are added to the messenger RNA to make it work properly within the cell. The cap and the tail will help that messenger RNA leave the nucleus the way it's supposed to. It will prevent it from being attacked by enzymes in the cytoplasm that are meant to cut up and break up stray pieces of RNA, and then it also helps the ribosomes bind to the messenger RNA. So here we have the DNA, which has these coding sequences in there, the introns in between the exons. Once the RNA is made and the cap and the tail are added, then there are enzymes that basically cut out the introns and then paste the exons back together to make the edited sequence of messenger RNA that can then leave the nucleus and go out into the cytoplasm. Once the messenger RNA gets out to the cytoplasm, it depends on another kind of messenger or another kind of RNA to help translate the sequence into a protein. Transfer RNA or tRNA is basically the language interpreter. These molecules pick up the appropriate amino acid, and then they have an anticodon that is complementary to the codon that allows them to put it in the proper place on the surface of the ribosome where the proteins are going to be made. Here's a diagram of a transfer RNA molecule. You see it's a kind of a partial partially twisted up like this, but at the end, at one end, it has the anti-codon that is complementary to the codon on the messenger RNA, and then at the other end, it's got a place to attach to the amino acid that it codes for. There's an enzyme that matches these up together, of course, but this will allow them to put them into place where it needs to go. So ribosomes are involved in the building of the polypeptides, and ribosomes are also made of RNA, ribosomal RNA, as well as some proteins that are attached to it, and the translation event occurs on the surface of the ribosome. There are two subunits of the ribosome, the small subunit and the large subunit, and they come together during that translation, and on the ribosome, in the ribosome, are the binding sites for messenger RNA and transfer RNA. So here we have a ribosome diagram. Okay, the larger subunit has the transfer RNA binding sites, and then the messenger RNA binding site is located on the small subunit. First, there is an initiation of the coding that occurs in two steps. The messenger RNA molecule binds to the small subunit of the ribosome, and the first transfer RNA binds to the messenger RNA at that start codon. That's going to read AUG and code for methionine, and the transfer RNA has the anti-codon UAC that matches up or is complementary to AUG. Once that happens, the large ribosomal subunit joins the small one, and that causes the ribosome to begin functioning. That first tRNA is going to be in the P site, which is going to hold the polypeptide chain, and then the A site is there to receive the next transfer RNA. And so here's our start of our genetic message. This is our AUG, okay, and there's the end, and there's our cap and our tail. And then here we have the start codon getting into place there with this methionine attached, the large subunit coming into place, and there's methionine in the position on the P site as the ribosome is put together. Once that initiation is started, then the amino acids can be added one by one to the first amino acid. This process is called elongation. That's the adding of the amino acids to the growing polypeptide chain. Then elongation continues until the termination stage where the stop codon is reached. And then that's going to free the polypeptide from the transfer RNA, and the ribosome is going to separate back into its subunits. So here we have the whole process. Here's our codon recognition. All right, our anti-codon is going to match up here to the next codon on the messenger RNA as that tRNA gets into the A site. That forms a peptide bond between the polypeptide chain and the next amino acid. Then that releases the previous transfer RNA, and the current one moves over into the P site, opening up the A site for another one to join. And so the whole process continues until it reaches the stop codon. Now, if there's a change somehow in that sequence, that's going to cause something called a mutation. Sometimes mutations are just a single nucleotide pair that gets changed. Sometimes it could be a large area of the chromosome, depending on what kind of mutation it happens to be. Mutations in the gene, rather than just the chromosome, can be divided into two basic categories. They can be substitutions, or there can be insertions and deletions. Okay, substitutions are going to be replacing one nucleotide with another. Sometimes this doesn't have any effect at all. This is called a silent mutation. In a case like this, it changes the codon to a different codon for the same amino acid, and so it doesn't cause any change in the protein sequence. Another one could change the amino acid coding, so putting in a different amino acid that would make a different protein, or the protein would be changed a little bit. And then another thing could be an improved protein that could occur, and sometimes it could change an amino acid into a stop codon. If it stops before the protein is finished, then you're going to have a malformed or misfunctional protein. Sometimes changing the amino acid can change the shape of the protein enough to cause a problem. or sometimes it's something that's improved. So that would be a beneficial mutation. Another kind of mutation would be a deletion or an insertion. And this is much more harmful overall because when you delete or insert an a codon or an amino acid in the polypeptide chain, it's going to alter the reading frame of the messenger RNA. That's going to group the codons differently, and that's going to lead to big changes in the sequence of amino acids downstream from that mutation. And definitely is almost always going to produce either a non-functional polypeptide or something that has a different function than what the original intent of the protein was. Here we have a substitution. So here's my normal hemoglobin. Here, this is my normal hemoglobin DNA. Code CTT and my messenger RNA is GAA. If there's a substitution here of an A for a T there, that's going to make this code for a different amino acid. Instead of glutamine, it codes for valine, and that's what happens in sickle cell anemia. It changes there's one codon that gets changed, one base that gets changed, and it changes the protein enough to make the hemoglobin fold differently and cause sickle cell anemia. And we'll talk more about how that works later on. Here are a couple of other things that can happen. Okay, there's my normal gene up there at the top. If I have a substitution of this guanine for an adenine, that changes this amino acid from glycine to serine. If we take out one of these uracils here and shift everything down that way, that changes everything after the lysine to a different amino acid, or if we insert something there in between the U and the G, then that's going to, or in between two U's here, then that would change everything from that point on as well, all of which are going to cause some kind of a problem with the eventual polypeptide.